So I will talk about gene regulation in prokaryotes, where prokaryotes, for example, bacteria, and the eukaryotes. So let me start with the regulation in bacteria. We tend to think of bacteria as simple, but even the simplest bacterium has complex tags when it comes to gene regulation. So gene regulation is not a simple thing. The bacteria in your gut or between your teeth have genomes that contain thousands of different genes. Most of these genes encode proteins, each with its own role in a process such as fuel metabolism, maintenance of cell structure, and defense against viruses. Some of these proteins are needed routinely, while others are needed only under certain circumstances. So, the genes are not always, processed all the time. Some of them is continuously read, but some of them depends on the environment, depends on the conditions that are need to be processed. Thus, cells don't express all the genes in their genome all the time. You can think of the genome as being like a cookbook with many different recipes in it. The cell will only use the recipes, expresses the genes, that means, that fit its current needs. There are various forms of gene regulation, that is, mechanisms for controlling which genes get expressed and what levels. So gene regulation means when do the cell expresses the gene and how to control it. However, a lot of gene regulation occurs at the level of transcription. You know what the transcription is, and also translation from our earlier videos. Bacteria have specific regulatory molecules that control whether a particular gene will be transcribed into mRNA. Often, these molecules act by binding to DNA near the gene and helping or blocking the transcription enzyme RNA polymerase. Let's take a closer look at how genes are regulated in bacteria. In bacteria, related genes are often found in a cluster on the chromosome where they are uh, transcribed from one promoter, okay, RNA polymerase binding site as a single unit. Such a cluster of genes under control of a single promoter is known as operon. So operons are common in bacteria, but they are rare in eukaryotes such as humans. In general, an operon will contain genes that function in the same process. For example, a well-studied operon called lag operon contains genes that encode proteins involved in uptake and metabolism of a particular sugar, lactose. So lag operon is responsible for this. Operons allow the cell to efficiently express sets of genes whose products are needed at the same time. Operons are aren't just made up of coding sequences of genes. Instead, they also contain regulatory DNA sequences that control transcription of the operon. Typically, these sequences are binding sites for regulatory proteins, which control how much the operon is transcribed. The promoter or site where RNA polymerase binds is one example of a regulatory DNA sequence. Most operons have other regulatory DNA sequences in addition to the promoter. These sequences are binding sites for regulatory proteins that turn expression of the operon up or down. Some regulatory proteins are repressors that bind to pieces of DNA called operators. When bound to its operator, a repressor reduces transcription. It reduces. For example, by blocking RNA polymerase from moving forward on DNA. Some regulatory proteins are activators. When an activator is bound to its DNA binding site, it increases transcription of the operon. For example, by helping RNA polymerase bind to the promoter. Where do the regulatory proteins come from? Okay, so that's the question. Like any other protein produced in an organism, they are encoded by genes in the bacterium's genome. The genes that encode regulatory proteins are sometimes called regulatory genes. Many regulatory proteins can themselves be turned on or off by specific small molecules. The small molecule binds to the protein, changing its shape and altering its ability to bind DNA. Okay, so for instance, an activator may only become active, able to bind DNA, when it is attached to a certain small molecule. Some operons are usually off, but can be turned on by a small molecule. The molecule is called inducer. Okay, and the operon is said to be inducible. For example, the lag operon is an inducible operon that encodes enzymes for metabolisms of the sugar lactose. It turns on only when the sugar lactose is present and other preferred sugars are absent. The inducer in this case is allolactose, a modified form of lactose. We have a simulation that you are going to play or you are going to study these lag operon, and also as a homework. We will see in detail how these operations in a lag operon becomes. Other operons are usually on, but can be turned off by a small molecule. The molecule is called core processor. Okay, and the operon is said to be repressible. Okay, for example, the TRP operon is a repressible operon that encodes enzymes for synthesis of the amino acid tryptophan. This operon is expressed by default, but can be repressed when high levels of amino acid tryptophan are present. The core processor in this case is tryptophan. Okay. These examples illustrate an important point that gene regulation allows bacteria to respond to changes in their environment by altering gene expression and thus changing the set of proteins present in the cell. Many genes play specialized roles and are expressed only under certain conditions, as described above. However, there are also genes whose products are constantly needed by the cell to maintain essential functions. These housekeeping genes are constantly expressed under normal growth conditions, constitutively active. It means housekeeping genes are have promoters and other regulatory DNA sequences that ensure constant expression. Okay, so let's talk about this gene regulation in eukaryotic cells. So our amazing body contains, you know what the eukaryotic cells, that cells that have a certain, you know, cell organelles covered with some membranes, etc. And human cells are one of them. So the body contains hundreds of different cell types, from immune cells to skin cells to neurons. Almost all of our cells contain the same set of DNA instructions, right? So why do they look so different? For example, this is how the neuron cell looks like and its structure, etc. And on the other hand, this is the liver cell. Structurally, they are very different. Although they contain the same set of information inside the nucleus, their appearance and their function, everything is completely different. So, what is the question? So, why is that so? The answer to the question why they look so different, because different gene regulation. So these genes are contained in all different kinds of cells, but they have different regulations. So this is gene regulation. Gene regulation is how cell controls which genes out of the many genes in its genome are turned on or expressed. Thanks to gene regulation, each cell type in our body has different set of active genes, despite the fact that almost all the cells of your body contain the exact same DNA. So these these different patterns of gene expression cause your various cell types to have different sets of proteins, making each cell type uniquely specialized to do its job. For example, one of the jobs of the liver is to remove toxic substances like alcohol from the bloodstream. To do this, liver cell express genes encoding subunits or pieces of an enzyme called alcohol dehydrogenase. Okay? This is an enzyme. This enzyme breaks alcohol down into non-toxic molecule. So this is what happens when you take alcohol. The liver reacts to it just by breaking the molecules of the alcohol. The neurons in person's brain don't remove toxins from the body, so they keep these genes unexpressed or turned off. Similarly, the cells of the liver don't send signals using neurotransmitters, so it's not its job. So they keep neurotransmitter genes turned off, so that they don't have these axons, dendrites, etc. There are many other genes that are expressed differently between liver cells and neurons, or any two cell types in a multicellular organism like ourselves. Now there is a tricky question. Many factors can affect which genes a cell expresses. Different cell types express different sets of genes, as we saw above. However, two different cells of the same type may also have different gene expression patterns depending on their environment and internal state. Broadly speaking, Uh, we can say that a cell's gene expression pattern is determined by information from both inside and outside the cell. Examples of information from inside the cell: the proteins it inherited from its mother cell, whether its DNA is damaged, and how much ATP it has. Examples of information from outside the cell: the chemical signals from other cells, mechanical signals from the extracellular matrix, and nutrient levels, as an example. So how do these cues help a cell decide what genes to express? Cells don't make decisions in the sense that you or I would. Instead, they have molecular pathways that convert information, such as binding of a chemical signal to its receptor, into a change in gene expression. As an example, let's consider how cells respond to growth factors. A growth factor, you see over here, a growth factor is a chemical signal from a neighboring cell that instructs a target cell to grow and divide. We could say that the cell notices the growth factor and decides to divide. But how do these processes actually occur? The cell detects the growth factor through physical binding of the growth factor to a receptor protein on the cell surface. Binding of the growth factor causes the receptor to change the shape, triggering a series of chemical events in the cell that activate proteins called transcription factors. The transcription factors bind to certain sequences of DNA in the nucleus and cause transcription of cell division related genes. The products of these genes are various types of proteins that make the cell divide, drive the cell growth, and or push the cell forward in the cell cycle. So this is just one example of how a cell can convert a source of information into a change in gene expression. There are many others, and understanding the logic of gene regulation is an area of ongoing research in biology or biotechnology today. Growth factor signaling is complex and involves the activation of a variety of targets, including both transcription factors and non-transcription factor proteins. You can learn more about these growth factor signaling, which is put in the notes under this page of the presentation. So eukaryotic gene expression can be regulated at many stages. In the articles that follow, I will examine different forms of eukaryotic gene regulation. That is, we'll see how the expression of genes in eukaryotes like us can be controlled at various stages, from the availability of DNA to the production of mRNAs, to the translation and processing of proteins. Eukaryotic gene expression involves many steps, and almost all of them can be regulated. Different genes are regulated at different proteins, and it's not uncommon for a gene, particularly an important or powerful one, to be regulated at multiple steps. One of them is chromatin accessibility. The structure of chromatin, DNA, and its organizing proteins can be regulated. More open or relaxed chromatin makes a gene more available for transcription. Transcription is a key regulatory point for many genes. Sets of transcription factors proteins bind to specific DNA sequences in or near a gene and promote or repress its transcription into RNA. And RNA splicing, RNA processing, splicing, capping, and addition of a poly A tail to an RNA molecule can be regulated and so can exit from the nucleus. Different mRNAs may be made from the same pre-mRNA by alternative splicing. The lifetime of an mRNA molecule in the cytosol affects how many proteins can be made from it. Small regulatory RNAs called miRNAs can bind to target mRNAs and cause them to be chopped up. Translation of an mRNA may be increased or inhibited by regulators. For instance, miRNAs sometimes block translation of their target mRNAs rather than causing them to be chopped up. Proteins can undergo a variety of modifications, such as being chopped up or tagged with the chemical groups. These modifications can be regulated and may affect the activity or behavior of the protein. Although all stages of expression can be regulated, the main control point for many genes is transcription. Later stages of regulation often refine the gene expression patterns that are roughed out during transcription. Differences in gene regulation makes the different cell types in multicellular organism, such as yourself, unique in structure and function. If we zoom out a step, gene regulation can also help us explain some of the differences in form and function between different species with relatively similar gene sequences. For instance, humans and chimpanzees have genomes that are about 98.8% identical at the DNA level. The protein coding sequences of some genes are different between humans and chimpanzees, contributing to the differences between the species. However, researchers also think that changes in gene regulation play major role in making humans and chimps different from one another. For instance, some DNA regions that are present in the chimpanzee genome but missing in the human genome contain known gene regulatory sequences that control when, where, or how strongly a gene is expressed. So, to give the summary, what are the key points so far that I try to explain? Gene regulation is the process of controlling which genes in a cell's DNA are expressed, used to make a functional product such as protein. Different cells in a multicellular organism may express very different sets of genes, even though they contain the same DNA. The set of genes expressed in a cell determines the set of proteins and functional RNAs it contains, given its unique properties. In eukaryotes like humans, gene expression involves many steps, and gene regulation can occur at any of these steps. However, many genes are regulated primarily at the level of transcription. Bacterial genes are often found in operons. Genes in an operon are transcribed as group and have a single promoter. By the way, we should know how the bacteria genes are regulated, because we have lots of bacteria in our body that has some, doing some functions for us. Each operon contains regulatory DNA sequences which act as binding sites for regulatory proteins that promote or inhibit transcription. Regulatory proteins often bind to small molecules, which can make the protein active or inactive by changing its ability to bind DNA. Some operons are inducible, meaning that they can be turned on by the presence of a particular small molecule. Others are repressible, meaning that they are on by default but can be turned off by a small molecule.